Lecture 2, Tools in Studying Microbiology. The 5 Eyes of Microbiology. 1. Inoculation. Introduction of a sample into a sterile culture media. 2. Incubation. Allowing cultures to grow under favorable growth conditions using an incubator. 3. Isolation. Separating one bacterial species to another using streak plating or pore plating techniques. 4. Inspection. Observation of different characteristics by naked eye, macroscopic, or under a microscope, microscopic. 5. Identification. Identifying the genus and species of the sample using biochemical tests, DNA-based methods, and immunological testing. Learning objectives. 1. Describe the basic principles of microscopy. 2. Differentiate various microscopic instruments used in microbiology. 3. Explain the principles of gram staining and other staining techniques. 4. Familiarize with tools used in bacterial identification. The microscope. Microscope. Instrument used to visualize micrometers, or micron-size objects, such as microorganisms, and cells, made up of lenses for magnification. Magnification, ability to enlarge objects. Resolving power, ability to show detail. Total magnification is the power objective times the power of ocular lens. Magnification in most microscopes results from interaction between visible light waves and curvature of the lens. Refraction, angle of light passing through convex surface of glass changes. Oil immersion lens requires the use of oil to prevent refractive loss of light. Types of microscopes. Light microscope uses visible light 400 to 750 nanometers. Magnification is between 40 to 2000 times. Electron microscope uses electron beams. Electron waves are 100,000 times shorter than the waves of visible light. Magnification is between 5,000 to 1,000,000 times. Parts of a microscope. Eyepiece, also known as the ocular. This is the part used to look through the microscope. Its standard magnification is 10 times, with an optional eyepiece having magnifications from 5 times to 30 times. Objective lenses. These are the major lenses used for specimen visualization. They have a magnification power of 40 times to 100 times. There are about 1 to 4 objective lenses placed on one microscope. Each lens has its own magnification power. Nose piece, also known as the revolving turret. It holds the objective lenses. It is movable, hence it can revolve the objective lenses depending on the magnification power of the lens. Adjustment knobs. These are knobs that are used to focus the microscope. There are 2 types of adjustment knobs: fine adjustment knobs and coarse adjustment knobs. Stage. This is the section in which the specimen is placed for viewing. They have stage clips that hold the specimen slides in place. The most common stage is the mechanical stage, which allows the control of the slides by moving the slides using the mechanical knobs on the stage instead of moving them manually. Aperture. This is a hole on the microscope stage through which the transmitted light from the source reaches the stage. Microscopic illuminator. This is the microscope's light source located at the base. It is used instead of a mirror. It captures light from an external source of a low voltage of about 100 volts. Condenser. These are lenses that are used to collect and focus light from the illuminator into the specimen. They are found under the stage next to the diaphragm of the microscope. They play a major role in ensuring clear, sharp images are produced with a high magnification of 400 times and above. The higher the magnification of the condenser, the more the image clarity. Diaphragm. It is also known as the iris. It is found under the stage of the microscope, and its primary role is to control the amount of light that reaches the specimen. It's an adjustable apparatus, hence controlling the light intensity and the size of the beam of light that gets to the specimen. Condenser focus knob. This is a knob that moves the condenser up or down, thus controlling the focus of light on the specimen. Stage control. It controls how far the stages should go, preventing the objective lens from getting too close to the specimen slide, which may damage the specimen. It is responsible for preventing the specimen slide from coming too far up and hitting the objective lens. Types of light microscopes. Bright field. Most widely used. Specimen is darker than the surrounding field. Live and preserved stained specimens. Dark field. Brightly illuminated specimens surrounded by dark field. Live and unstained specimens. Phase contrast. Transforms subtle changes in light waves passing through the specimen into differences in light intensity. Best for observing intracellular structures. Differential interference contrast or Nomerski interference contrast. Uses interference patterns to enhance contrast between different features of specimens. Useful in distinguishing structures within live specimens. No staining. Fluorescence. Uses fluorescent dyes that emit visible light when bombarded with shorter UV rays. Can distinguish between living and dead cells. Confocal laser scanning. Uses laser beam of light that scan at various depths in the specimen. Types of electron microscopes. Transmission electron microscope, TEM. Transmit electrons through the specimen. Darker areas represent thicker, denser parts, and lighter areas indicate more transparent, less dense parts. Can resolve subcellular structures, such as organelles, and interior of membranes, and molecular structures, such as DNA. Cells are embedded in a plastic resin and dehydrated using different concentrations of ethanol. Thin sections, about 20 to 100 nanometers thick, are cut using ultramicrotome. Samples are fixed to copper wire or carbon fiber grids and stained with uranyl acetate or osmium tetroxide, which contain electron-dense heavy metal atoms. Scanning electron microscope, SEM. Provides detailed three-dimensional view. Bombards the surface of a whole, metal-coated specimen with electrons while scanning back and forth over it. Can resolve surfaces of larger specimens like bacterial cells. Samples are mounted on a wire grid and sputter coated with gold or palladium before viewing. Ultramicrotome used for the preparation of ultra-thin sections of samples for TEM. Principles of staining techniques. Stains or dye. Salts composed of negative or positive ion, one of which is colored the chromophore. Dyes create contrast by imparting a color to cells or cell parts. Basic dyes with positive charges on the chromophore binds with negatively charged surface. Examples: methylene blue, Still violet, safranin. Acidic dyes, with negative charges on the chromophore. Binds with positively charged surface. Example, India ink or Inoculation. Positive staining. Surfaces of microbes are negatively charged due to teichoic acid, which attract basic dyes. Negative staining. Microbe repels dye, the dye stains the background. Types of staining. Simple stains. One dye is used. Reveals shape, size, and arrangement. Differential stains. Use a primary stain and a counter stain to distinguish cell types or parts. Gram stain. Differentiates between gram-positive and gram-negative bacteria. Dyes. Crystal violet, safranin. Acid-fast stain or Zeil-Nielsen stain. Differentiates cells with mycolic acid present in the cell wall of Nocardia and mycobacteria. Dye. Carbalfusin. Endospore stain. Uses heat to drive the primary stain into the endospore. Dye. Malachite green. Special stains. Reveal certain cell parts not revealed by conventional methods. Capsule stain. Uses acidic dyes like eosin and nigrosin, which stains the background and leave the cells colorless. Flagellar stain. Stain, such as carbalfusin, and mordant, such as tannic acid, can be used to increase the diameter of the flagella. Gram staining principle and procedure. Developed by Hans Christian Gram. A differential staining technique which uses more than one dye. Crystal violet as primary stain, and safranin as the secondary stain, which distinguishes gram-positive and gram-negative bacterial cells. It uses iodine solution as a mordant, which forms insoluble complex with the crystal violet stain, and increases its affinity to the thick peptidoglycan layer of gram-positive bacteria. During the decolorizing step, alcohol dissolves the lipids in gram-negative cell walls and increases its permeability, allowing the crystal violet iodine complex to be washed out of the cell. The secondary stain, safranin, will be absorbed by the gram-negative bacterial cells in the final step, which will appear red. Gram-positive bacterial cells will retain the crystal violet stain, which will appear purple. Specimen preparation for optical microscopes. Wet mounts and hanging drop mounts allow examination of characteristics of live cells, such as motility, shape, and arrangement. Fixed mounts are made by drying and heating a film of specimen. This smear is stained using dyes to permit visualization of cells or cell parts. Fixation, process by which the internal and external structures of the cells are preserved and fixed in position. Heat fixation, by gently flame heating. Chemical fixation is used to protect fine cellular structures using ethanol, acetic acid, formaldehyde, or glutaraldehyde. Tools used in bacterial identification. Biochemical tests. Tests used for the identification of bacterial species based on the differences in the biochemical activities. Examples: starch hydrolysis test, used to observe amylase production in a bacterial culture indicated by a clearing zone around the colonies. Lipid hydrolysis test, used to test ability of bacterial culture to hydrolyze lipids. Positive reaction is observed by the formation of clearing zone. Gelatin hydrolysis test. Used to identify bacteria, which can produce extracellular proteolytic enzymes, or gelatinases, that liquefy gelatin, an animal protein. Positive reaction is observed from the liquefaction of the nutrient gelatin medium. Analytical Profile Index, API, is a miniaturized panel of biochemical tests compiled for identification of groups of gram-negative bacteria. The kit contains 20 miniature biochemical tests for manual identification up to the species level. Bacterial suspension is used to rehydrate each well, and the strips are incubated. During incubation, metabolism of the substrate produces color changes, which will indicate positive or negative reaction. All positive and negative reactions are compiled to generate a profile number. The 7-digital code obtained from the reaction results is used to identify the sample using the API catalog or API web online. Biolog Microbial ID allows identification of gram-positive and gram-negative bacteria in a single panel. It analyzes the ability of the cell to metabolize all major biochemicals, at the same time determining other important physiological properties such as pH, salt, lactic acid tolerance, reducing power, and chemical sensitivity by creating a metabolic fingerprint from discrete test reactions performed within a 96-well-clear plate. Culture suspensions are tested with a panel of pre-selected assays, then incubated, read, and compared to extensive databases of environmental organisms, human pathogens, animal and plant pathogens. Analysis of nucleic acids. DNA hybridization. When two organisms share many identical or highly similar genes, their DNAs are expected to hybridize. DNA hybridization, therefore, is useful for differentiating between organisms as a complement to small subunit rRNA gene sequencing. GC ratios. The GC ratio is the percentage of guanine plus cytosine in an organism's genomic DNA. If two organisms' GC ratios differ by more than about 5%, they have few DNA sequences in common, and therefore unlikely to be closely related.